Welcome to the Deep Dive. Today we're plunging into the world of bioinformatics.
Sounds exciting.
Yeah. Now that term, bioinformatics, it might sound a bit intimidating. Maybe you're picturing someone battling lines of code.
Hey, yeah, I get that. Well, there's some truth to the coding parts sometimes.
Right.
But really, at its heart, bioinformatics is about using computers, using their power to make sense of the incredibly complex data we get from studying living things.
Okay, so like a tool to understand life's molecules without needing a PhD in computer science overnight.
Exactly. Think of it like that, a way to get at the fundamentals.
So our mission today, what are we focusing on?
Right. So for this deep dive, we want to explore two key areas where these computational tools are making, well, just huge strides.
Okay.
First, how bioinformatics helps us analyze RNA, those really vital messengers inside our cells. And second, how it lets us predict the incredibly intricate 3D shapes of proteins, you know, the tiny machines doing all the work.
Proteins and RNA, core stuff.
Absolutely fundamental biological processes. And bioinformatics is giving us totally new ways to, investigate them.
Okay, let's unpack that a bit. I mean, think about how much information is packed into our genes, right? Our DNA.
It's staggering.
So bioinformatics gives us the tools to decode all that and figure out how it's actually used to build and run an organism.
That's the core idea. And studying RNA is a really powerful way we do that.
Right. RNA. Tell us about that.
Okay, so a key technology here is RNA sequencing, usually called RNA-seq.
RNA-seq. Got it.
What's really remarkable about RNA-seq is that it lets us do two things at once. Discover and measure pretty much all the RNA molecules in a sample at a specific time.
So discovery and counting, basically.
Exactly. It lets us see which genes are active, switched on, and crucially, how active they are. What's the level?
Okay. So not just on-off, but the volume control too.
Precisely. And that ability to see which genes are up or down in different states, say, healthy versus diseased, that's just a fundamental shift.
Yeah.
We're moving beyond the DNA blueprint to see what's actually happening functionally.
It sounds like this isn't some niche technique anymore. It must be pretty widespread now.
Oh, absolutely. It's become a really essential tool for loads of different biological research. Its use has spread way beyond just genomics, folks.
So anyone studying life sciences might use it?
Pretty much. If you're trying to understand how genes are expressed, how they're regulated, RNA-seq gives you that direct snapshot, the functional output of the genes.
Okay, now here's something that caught my eye. I read there isn't just one single recipe for analyzing RNA-seq data.
That's right. It really depends.
Depends on what?
Well, mainly on the organism you're studying and the specific questions you're asking.
Can you give us an example or two?
Sure. So a big factor is whether we have a complete map of the organism's genes, its genome.
Okay, like the Human Genome Project map.
Exactly. If we have that reference genome, we can take all the RNA sequences we get from RNA-seq, these are called reads, and map them back to their original positions on the genome.
Like finding where they came from on the big map.
Precisely. It helps us identify the exact RNA molecules, the transcripts. But there's a twist.
Oh.
Remember, RNA is often processed. Bits called introns are removed. So when you map the RNA read back to the DNA genome sequence, there might be gaps where those introns used to be.
Okay. The RNA sequence doesn't perfectly match the DNA sequence in one continuous stretch.
Right, so we need special alignment tools, we call them gapped aligners, that can handle these jumps across the intron regions.
Got it. Like allowing for missing words when fitting a sentence back onto a page. That makes sense. But what if you don't have that nice genome map, say, for a less studied organism?
Good question. That's a different challenge. It's more like, well, like trying to piece together a conversation you only had snippets of without knowing the topic or even who was talking.
Sounds tricky.
It is. The process is called de novo transcript reconstruction. De novo meaning from scratch.
So you build the transcripts from the fragments themselves.
Exactly. We take all the RNA sequence reads and try to computationally assemble them into longer, hopefully complete, transcript sequences. Then we can study those assembled sequences.
Does having more data help there?
Generally, yes. More reads and longer reads definitely help get a more complete picture. But interestingly, sometimes too much data can actually make the assembly computationally harder, create ambiguities.
Oh, okay. So there's a sweet spot.
Kind of. And if you're comparing different samples, say, treated versus untreated cells, it's often best to pool all the reads from all samples together first, do the assembly on that combined set to create a good reference, then map reads from each sample back to that reference to compare them.
Okay, so even without a genetic blueprint, you can still build one from the RNA messages themselves. That's quite powerful.
It really is.
And what about for organisms where we do have a really well-studied genome, like humans?
Yeah.
Does RNA-seq still tell us new things?
Oh, absolutely. Even with a detailed genome map, RNA-seq is hugely valuable. For one, it can help us find completely new RNA transcripts that weren't previously annotated in the genome, things we didn't know existed.
Discovering new stuff.
Yep. And maybe even more importantly, it lets us see how the activity levels of known genes change in different situations. This is what we call differential gene expression.
Comparing activity levels between conditions.
Exactly. Compare healthy versus diseased tissue or before and after a drug treatment. Pinpoint which genes change their expression level significantly. That dynamic view is crucial for understanding disease mechanisms, development, responses, you name it.
Yeah, I can see that being incredibly useful for things like cancer research or figuring out how our bodies fight off a new virus.
Precisely.
Now, if you're doing these big experiments, maybe with lots of samples,
Yeah.
I imagine you have to be pretty careful in how you set them up to make sure the results are solid.
Oh, absolutely critical. Good experimental design is paramount for RNA-seq, especially with many samples.
Like what?
Well, you definitely need proper controls, baselines to compare against, and randomization is key. Randomizing how you process samples helps avoid unintentional biases creeping in and messing up your results. You don't want batch effects, for example.
Right, making sure any differences you see are real, not just because you processed one group on Monday and the other on Tuesday.
Exactly that. You have to account for that potential variation.
Okay, so let's say we've done our homework, designed a good experiment, got our controls, randomized, and now we have this mountain of RNA-seq data.
Huh?
What are the main steps to actually analyze it? High level.
Okay, high level. Step one is always quality control. Check the raw sequencing data. Is it good quality? Are there errors we need to deal with?
Makes sense. Garbage in, garbage out.
Pretty much. Then comes read alignment, figuring out where those RNA sequence reads came from.
Which we talked about, either mapping to a genome, possibly with gaps.
Right, using those gapped aligners. Or sometimes you might align reads to a known set of transcript sequences directly to transcriptome. In that case, you might use ungapped aligners because you expect the read to match a transcript sequence more continuously.
Okay, genome or transcriptome alignment. Got it. What's next?
Quantification. This is where you actually count or measure how much of each gene or transcript is present in each sample. How many reads mapped to gene A versus gene B.
So getting the actual numbers.
Yep. And then finally, if your goal is comparison, you do differential gene expression analysis, using statistical methods to find genes whose quantified levels change significantly between your experimental conditions or groups.
Finding the important changes.
That's the goal.
That gapped versus ungapped alignment makes more sense now, thinking about those introns missing from the RNA. And I know RNA isn't always simple transcripts from genes. What about things like alternative splicing, where one gene makes different RNA versions, or those small regulatory RNAs?
Yeah, great point. RNA-seq is versatile enough to help study those too. Well, by carefully analyzing how the reads map, especially across exon junctions, we can identify different splicing patterns, alternative splicing. We can see which versions of an RNA are being made.
So you can see if a cell is choosing to make version A or version B of a protein from the same gene.
Exactly. And it can also detect things like fusion transcripts, where RNA is made from bits of two different genes joined together, sometimes seen in cancer.
Wow, okay.
And yes, it's also crucial for studying small RNAs, like microRNAs. These are tiny RNA molecules, but they're powerful regulators of other genes. Specific protocols and analysis tools exist for those.
So RNA-seq gives this incredibly detailed picture of all the different RNA molecules buzzing around in a cell. How does that then connect to the bigger picture, like understanding gene function?
Right. This leads into what we call functional profiling.
Functional profiling.
Yeah. By seeing which genes are active together or which genes go up or down in response to something, we can start to infer the biological processes or pathways that are being affected.
So if a whole bunch of genes related to, say, immune response are suddenly very active.
It gives you a strong hint that an immune response pathway is activated in that sample. You can use databases of known pathways to see which ones are enriched in your set of differentially expressed genes.
Okay, connecting the gene list to actual biological functions.
So that's the idea.
And it sounds like RNA-seq isn't just a standalone thing. It can be combined with other ways of studying the cell. to get an even richer picture.
Absolutely. Integration is really powerful, combining omics, as they say.
Omics?
Yeah. Genomics, transcriptomics, which is RNA-seq, proteomics, studying different layers of biological information.
Okay, so what can you combine RNA-seq with?
Well, for instance, you can combine it with studying genetic variations like SNPs, those single-letter changes in DNA.
Right.
By correlating SNP patterns with gene expression levels across many individuals, you can find variations that influence how much a gene is expressed. These are called EQTL's expression, quantitative trait loci.
Linking genetic differences to functional differences in gene activity.
Exactly. It helps connect genotype to phenotype at the molecular level. RNA-seq can even be sensitive enough sometimes to tell you if the RNA is coming from the copy of the gene inherited from the mother versus the father.
Whoa, really? You can see allele-specific expression.
Yes, under the right conditions and with the right analysis. It's pretty amazing.
That is incredible. What else? What other technologies pair well?
Well, if you want to understand how gene expression is controlled, the mechanisms,
Yeah.
you can combine RNA-seq with techniques like ChIP-seq. ChIP-seq tells you where specific proteins, like transcription factors, the master switches for genes, are binding to the DNA.
Okay, so where the switches are physically located.
Right. Or where certain chromatin features are, that's how the DNA is packaged, which affects accessibility. Combining that with RNA-seq helps you understand the regulatory logic, what turns a gene on or off.
Seeing the cause and effect, maybe?
Getting closer to it, yeah. And we mentioned microRNAs. You can combine RNA-seq, measuring the target mRNAs, with miRNA, measuring the microRNAs themselves, to understand those regulatory relationships.
Trying to figure out which microRNA controls which messenger RNA.
Exactly. Though predicting microRNA targets accurately is still tricky. It's usually best practice to use predictions from several different tools and look for consensus.
Okay, rely on multiple lines of evidence.
Good strategy. And then a really huge development is single-cell RNA-seq.
Ah, yes, I've heard about that. Looking at individual cells.
Exactly. Instead of averaging the RNA levels across thousands or millions of cells in a tissue sample.
Which could hide a lot of variation.
Totally. Single-cell RNA-seq lets you measure the RNA profile of each individual cell. This gives you incredible resolution to see different cell types or different states within a population that you'd completely miss with bulk RNA-seq.
Wow, it's like zooming in from a blurry crowd photo to seeing individual faces.
That's a great analogy. You see the heterogeneity, the nuances. It's transforming fields like developmental biology and immunology.
Okay, it feels like each technology adds another layer, another piece of the puzzle. Put them together and you get this incredibly detailed view inside the cell.
That's the power of integrative bioinformatics.
All right, so we've covered RNA, the messengers. Let's switch gears now to the workhorses, proteins.
The molecular machines.
Exactly. And I know that understanding their 3D shape, their structure, is super important for figuring out what they actually do.
It's absolutely fundamental, yeah. A protein's 3D structure dictates its function.
Like the key and lock idea.
Precisely. The specific shape allows it to bind to other molecules, catalyze reactions, form cellular structures, whatever its job is.
Okay. But here's the thing. We know the sequences, the amino acid chains, for billions of proteins, right? From all the genome sequencing.
Billions, yes. The sequence databases are enormous.
But the number of proteins where we actually know the 3D structure, determined experimentally, that number is much, much smaller, isn't it?
Vastly smaller. Maybe around a couple 100,000 structures in the protein data bank, the PDB, compared to billions of sequences. It's a huge gap.
Why is there such a big gap? Is it hard to figure out the structure?
Extremely hard. Determining protein structures experimentally, usually using techniques like X-ray crystallography or cryo-electron microscopy. It's painstaking work. It can take months, often years, of dedicated effort for just one protein. It's slow, expensive, and doesn't always work.
Okay, so a major bottleneck. We have the parts list, the sequence, but not the assembly instructions or the final shape for most proteins.
That's a good way to put it.
So how does bioinformatics step in here? How do we bridge that massive gap?
This is where computational protein structure prediction comes into play.
Predicting the shape from the sequence using computers.
That's the holy grail. The goal is to accurately predict the 3D shape just from the amino acid sequence. This is famously known as the protein folding problem.
Protein folding problem. I've heard that phrase. It's challenging.
Oh, it's been one of the grand challenges in biology for over 50 years. How does a linear chain of amino acids reliably fold up into a specific functional 3D structure?
50 years. Wow. So what approaches have scientists tried over the decades?
Well, historically, there have been two main camps, broadly speaking.
All right.
The first is physics-based methods, trying to simulate the actual the forces between atoms that cause the protein chain to fold.
It sounds logical, like simulating gravity making a ball roll downhill.
Kind of, but the forces are incredibly complex, and proteins are big molecules. Simulating this accurately requires immense computational power, huge amounts of computer time. It's been very, very difficult to get right, especially for larger proteins.
Okay, so physics is hard to simulate accurately at that scale. What's the other approach?
The other approach relies more on evolutionary information and pattern recognition.
How does that work?
The core idea is that proteins with similar functions or that evolved from a common ancestor often have similar sequences and, crucially, similar 3D structures.
Okay, evolution reuses successful designs.
Exactly. So if you have a protein whose structure you want to predict, you look for related proteins with known structures. If you find one, a template, you can use its structure as a starting point. That's called homology modeling.
Makes sense. If my protein looks like one we already know the shape of, maybe its shape is similar too?
Right. But what if there are no known related structures? That's where it got really tricky. But even then, you could look at the patterns within the sequence family of your protein.
How does that help?
If you align the sequences of many related proteins, say, the same protein from hundreds of different bacteria, you can see which amino acids tend to change together during evolution. If two amino acids far apart in the sequence chain consistently mutate together, it's a strong hint they might be close together in the 3D folded structure, interacting somehow.
Correlated mutations hinting at spatial proximity. Clever.
Very clever. And the explosion of sequence data from genome projects massively increased the amount of information available for this kind of analysis. Plus, advances in machine learning, especially depth learning, have really supercharged this evolutionary approach in recent years.
And I guess this is where AlphaFold comes in. It's been making massive headlines.
Exactly. AlphaFold, developed by DeepMind, really represents a paradigm shift, a major breakthrough.
What makes it so special?
It's a deep learning-based method that can predict protein structures with often near experimental accuracy, even in many cases where there are no similar known structures to use as templates.
Near experimental accuracy. That's huge.
It is. Its performance was stunningly demonstrated in the CASP competitions.
Right.
That's the critical assessment of protein structure prediction, kind of the Olympics for structure prediction. AlphaFold just blew away the competition.
Wow. Okay, so what's under the hood? What are the key innovations that make AlphaFold work so well? Its secret sauce.
Well, it combines several really smart ideas, mostly centered around deep learning. First, it heavily incorporates existing knowledge about the physics and geometry of protein structures into the neural network architecture itself.
Okay, building in some rules.
Right. Second, and crucially, it leverages those multiple sequence alignments, or MSAs we talked about, looking at large families of related sequences to find those evolutionary clues.
Using the wisdom of evolution.
Exactly. Third, it uses a novel neural network architecture they developed called the evoformer. This block is really clever because it can simultaneously process information about the sequence itself and the relationships between pairs of amino acids, like how likely are amino acid 5 and amino acid 50 to be close in 3D space.
So it's thinking about both the sequence and the potential 3D interactions at the same time.
Precisely. It reasons about the structure as it processes the sequence information. And fourth, it has a specific structure module at the end that explicitly predicts the 3D coordinates of all the atoms in the protein, producing the final structure. It also iterates, refining the structure prediction multiple times.
Iterating? Like taking a guess and then improving it?
That kind of, yeah. They call it recycling. The output structure is fed back into the network to refine the prediction further.
Okay, conceptually, how does it think about this? You mentioned pairwise relationships.
You can think of it as viewing the protein structure prediction problem as a kind of graph problem in 3D space.
A graph.
Yeah, where each amino acid is a node in the graph, and the network tries to figure out the edges, the relationships, the distances, the orientations between all pairs of nodes to build the final 3D structure. The evoformer is key to learning and refining that graph representation.
That's a helpful way to picture it. And you said it relies heavily on those multiple sequence alignments, the MSAs. Where does it get all that sequence data from?
It uses powerful search tools like Jackhammer and HHBlitz to scan absolutely massive protein sequence databases, things like Uniclus 30, BFD, and importantly, MGenify.
MGenify, what's that?
That contains sequences from metagenomic projects, remember, genetic material sequenced directly from environmental samples like soil or seawater.
Right. So capturing sequences from organisms we haven't even cultured in the lab.
Exactly. And including this vast, diverse metagenomic data significantly boosts prediction accuracy, especially for proteins that don't have many close relatives in the standard databases. The more diverse sequences you feed it, generally the better the prediction.
So the sheer scale of sequence data we now have is fuel for AlphaFold's engine.
Absolutely critical fuel.
How does AlphaFold actually learn to do this? How is it trained?
It's trained primarily on the experimentally determined structures in the protein data bank, the PDB, learning the relationship between sequence and known structures.
Okay, learning from examples.
Yes, but it also uses a really interesting technique during training called self-distillation.
Self-distillation. Sounds like it's teaching itself.
That's pretty much it. The network makes predictions on a huge data set of sequences where the structure isn't known, unlabeled data. It then takes its own high-confidence predictions from this set and uses them as additional training examples to learn from.
Wow. So it bootstraps its own learning process using its own best guesses.
Essentially, yes. It helps it generalize better and improves accuracy, especially for proteins that are different from those in the PDB training set.
That's fascinating. And once AlphaFold spits out a predicted structure, is it just a black box? Or can scientists get some idea of how it arrived at that structure?
That's a good question about deep learning models. While they can be complex, AlphaFold was designed with some interpretability in mind. Researchers can actually look at the intermediate representations and predictions within the network, like those pairwise distance predictions from the evoformer block.
So you can peek inside a bit.
Yeah, you can see how the network's confidence and its picture of the structure evolved through the process, which can give insights into why it predicted a certain fold. It also provides confidence scores for different parts of the predicted structure, which is really useful.
Confidence scores. So it tells you which parts of the prediction it's more sure about.
Exactly, which is crucial for knowing how much to trust the prediction for downstream use.
Okay, let's connect this back.
Yeah.
You mentioned earlier that huge number of protein families with unknown structures, often found through things like metagenomics. How is AlphaFold and methods like it impacting our ability to finally get a handle on all those unknowns?
It's transformative for that exact problem. Remember, there are thousands of protein families, often defined by sequence similarity alone, like in the FAM database, where we had absolutely no structural information. This represented a massive dark matter of the protein universe.
This structural dark matter, I like that.
Yeah. Computational methods, initially things like Rosetta using evolutionary couplings, and now supercharged by AlphaFold and similar deep learning approaches, are finally letting us shine a light on this dark matter. We can now generate plausible structural models for a vast number of these previously unknown families.
So we can start to see the shapes of these mysterious proteins.
Yes. There was work, even predating AlphaFold's public release, but using similar principles combined with metagenomics that predicted reliable structures for hundreds of previously unknown protein families. Many of them revealed entirely new types of protein folds, shapes never seen before in the PDB.
Discovering new folds just through computation and sequence data.
Exactly. Again, the key was having enough diverse sequences within a family, often thanks to metagenomics, to allow the computational methods to reliably infer the structure. There's even a metric, NF, quantifying sequence diversity, that correlates well with prediction accuracy.
So the combination of massive sequencing and powerful prediction algorithms is really unlocking this structural diversity.
It really is. And these models aren't just pictures. They're starting points for understanding function.
Can you give a concrete example of how a computational model helped experimentally?
Yeah, there was a great example with a complex membrane protein called cytochrome bead oxidase. It's important in bacteria. The experimental data from cryo-EM was incomplete. They couldn't build the full structure from the map alone.
Okay, so the experiment hit a wall.
Right. But researchers used a computational model built using evolutionary coupling information similar to what AlphaFold uses, and that model fit perfectly into the experimental map, providing the missing information needed to complete and interpret the structure.
Wow, so the computational prediction directly helped solve the experimental structure.
Exactly. It shows how these approaches are becoming synergistic with experiments, not just replacing them. They guide experiments, help interpret tricky data, and allow us to model things that are currently impossible to solve experimentally.
So these tools aren't just about predicting static shapes, right? They're a foundation for more.
Absolutely. They provide the basis for understanding protein function, for interpreting the effects of genetic mutations. How does a mutation change the structure and thus the function? For designing new drugs that target specific protein shapes, and for guiding future experiments. It's really accelerating biological discovery.
So we've taken this pretty fascinating deep dive today, looking at how bioinformatics, these computational tools, are really revolutionizing how we understand both RNA, the gene messengers, and proteins, the cellular workhorses.
Yeah, it's quite amazing when you step back and see how computers are letting us tackle these incredibly complex biological questions.
It feels like we can get insights from this massive amount of biological data much, much faster now.
We definitely can. The speed and scale are just on a different level than purely experimental approaches could achieve alone.
So for everyone listening, the next time you hear about, a new gene discovery or a disease being studied at the molecular level, just remember that bioinformatics is almost certainly playing a huge role behind the scenes, helping scientists decode the fundamental building blocks of life.
Couldn't have said it better myself. It's deeply embedded in modern biology now.
And it leaves you wondering, with sequencing getting faster and cheaper, and computational methods like AlphaFold getting even better, what's next?
Right. What other biological mysteries, things that seemed totally intractable just a decade ago, might we be on the verge of unlocking now?
Yeah. Think about the potential for new medicines, for biotechnology, for just fundamentally understanding how life works. It's pretty exciting.
It really is. The future of biology is definitely computational.